Good afternoon, everyone. I'm the third presenter, Qingling, and my topic is the effects of high-pressure processing HPP on quality attributes of hard clams, Maretrix lusoriae. Chinese title: 高压处理对文革品质特性的影响。In my content, I will divide into 4 parts and start with introduction. The applications of HPP high-pressure processing in food processing was first investigated in 1899, and the process has been recognized as a non-thermal process technique for preservation of food for over a century. One of the principal advantages of the HPP process is the expanded shelf life and improvement of food safety due to the inactivation affected in microbial population. How powerful is HPP? As 5 tons of elephant stand above a coin, and it's about 400 megapar. 1 ATM equals to 0.1 megapar, therefore it's about 4,000 ATM. The applications of HPP in foods are various, and these are the examples. We have the fruit juice, shellfish, meats, fish, ready-to-eat meals, and dairy products. The use of HPP on bivalve mollusks is currently under study more and of interest due to its minimal effects on sensory characteristics and nutritional quality of this organism. This technology is used for shucking of oyster and other mollusks and has proven efficient in reducing microorganisms load, including of certain pathogens such as Vibrio parahemolyticus. Hard clams are widely found throughout Asia, Japan, Korea, China, Taiwan, and elsewhere as a species of saltwater clams, a marine bivalve mollusk in the family Venaridae, the Venus clam. They are one of the most important economic bivalves in Taiwan. Their natural habitat is offshore or upstream. Mollusks, particularly bivalves, are often associated with food safety issues due to the recurrence episodes of gastrointestinal infections and food poisoning. Center for Disease Control and Prevention (CDC) and public health and regulatory officials in several states investigated a multi-state outbreak of gastrointestinal illnesses linked to raw oyster harvest from Mexico in 2019. The victims in this outbreak were infected with multiple pathogens caused illness. These are the previous literature which using high pressure processing on the shellfish. From the previous literatures one shows that the HPP shucking effects on oyster. When using 311 megapar, they have 100% of full release of adductor muscle, which state that using higher pressures of HPP can increase the shucking effect. In previous literature, 2 shows the figure 3 effects of HPP on the appearance and volume of clams meat. The A is untreated sample and B is using 552 megapar for 3 minutes. The result showing using a higher pressure which can increase the volume and lighter in color. Next, the aim of this study is to determine the optimum HPP conditions for hard clams according to shucking effect, microbiological, chemical, and physical analysis, and to investigate the effect of different pressure use on hard clams during refrigerator storage. Materials and methods. the hard clams were under pretreatments, and then we will divide into two groups. The first, we were using the two different minutes, which is 3 and 5 minutes, and the pressure used was 200 to 600 megapascal, and the 0.1 is the control groups. After the treatments, we will have the microbiological, chemical, and physical analysis. Then we have 3 minutes as our storage experiment, and using the same pressure, which is 200 to 600 megapascal, and the control group is 0.1 megapascal. And sample at 4 degrees Celsius, the store at 4 degrees Celsius for 15 days, and sampling each 3 days. This is the analysis that we have done. For the pretreatments of hard clams, the fresh hard clams were bought from the market and soaked at 2% of salt water for room temperature at 2 hours. This step is to descending the clams and as a purification. Next, we will have to pack the clams separately in the polyethylene plastic bag and adding 9 ml of 1% salt water. Then we will pack into a bigger size of polyethylene plastic bag and under vacuum. Result and discussion. Figure 1 shows the image of hard clam meats after HPP treatment. As the result shows, we can see at 300 megapascal to 600 megapascal had a bigger size of the clam meats and expanded and also swollen when compared to 200 and the control groups. Then, when using the HPP treatment at 5 minutes, has a whiter appearance compared to the 3 minutes. Table 1 shows the LABW value of hard clam meats after the HPP treatment. And L represents brightness, A is red to green, B is yellow to blue, and W is whiteness. As the result shows, all the HPP treatment groups are higher than the control groups for the value L and W. And for the 5 minutes groups are higher than the 3 minutes groups in the value L and W. Next, when we focus on the value A and B, the result shows using HPP treatments has a lower value when compared to the control group. Table 2 shows the HPP shocking effects on oyster. Negative means no release of adductor muscles after HPP treatment, which the clams will, all the meat will stick to the shell. And next, we will have 0 means partial release of adductor muscle. There are still some of the adductor muscle which stick to the shell and not fully released. Then we will have the positive. Positive is full release of adductor muscle. As the image shows, we can see that the shell is clear without any of the meat. And from the result, we know that the HPP treatments using 400 megapascal and above can have the best shocking effect. Table 3 shows the moisture contents of hard clam meats after HPP treatment. As the moisture content will increase as the pressure increases. And also, when the 500 minutes time of HPP treatments are higher than the 3 minutes. The increase in moisture may be due to the increased water absorption by protein, which is consistent with earlier reports that HPP can increase the hydration of protein. Figure 2 shows the expanding rate of hard clam meats after HPP treatment. The different lower case indicates significant difference at the same time, while the upper case indicates no significant difference at the same pressure. As the result shows, the pressure increased with the increase of expanding rate, but there was no significant change between 3 minutes and 5 minutes. In previous articles also found the same result, which the increase in volume of the clam was most likely due to the infusions of the liquid into clam's tissue during pressurization. Figure 3 shows the aerobic leg counts of clam meats after HPP treatment. As the result shows, when the pressure increases, the value of APC will decrease. And when compared to 3 minutes and 5 minutes, there were only in 600 megapascal who have significant difference. Figure 4 shows the saprophytic bacterial count PBC of hard clam meats after the HPP treatment. This result was similar to APC, which the saprophytic bacterial count will decrease with the increase of pressure. And when compared to 3 and 5 minutes, there were only significant change in the 500 megapascal and 600 megapascal. Table 4 shows the value of coliform, E. coli, H2S producing bacteria, HPC, and viral parahemolyticus VP in the hard clam meats after the treatment. The coliform and VP were only detected in the control groups and 200 megapascal, while the HPC were only detected in control groups 200 megapascal and 300 megapascal for 3 minutes. And the E. coli was not detected for all the groups. This result showing using more than 400 megapascal can inactivate the growth of coliform, E. coli, HPC, and VP. Figure 5 shows the pH value of hard clam meats after HPP treatment. The range of the pH value for all the groups were in 6.14 to 7.14. There were a slightly increase of pH value for the pressure groups compared to the controls. Figure 6 shows the total volatile basic nitrogen TBBN of hard clam meats after HPP treatment. As a result showing in above, the pressure groups has increase of the TBBN when compared to the control groups. And the 3 minutes group has a higher value of TBBN compared to the 5 minutes. Although there was a change of TBBN for the pressure groups, but for the other groups will not exceed the regulatory standard, which is 25 mg for 100 grams. Figure 7 shows the change of aerobic leg counts in hard clam meats after HPP treatment during storage at 4 degrees Celsius. Left right represents 6.47 log CFU per grams of APC as the regulatory standard for frozen fish and shellfish. From the result showing for 0.1 megapascal and 200 megapascal, showing that at day 3, the level of APC has exceeded the regulatory standard. And the table shows the 300 megapascal, 400, 500, 600 megapascal the days that they can store. And 300 and 400 megapar are 3 days and 6 days, while the 500 megapar and 600 megapar, which have the longest, is 9 days. Figure 8 shows the change of cyclophilic bacteria count PBC in hard clam meats after HPP treatment during storage. From the results showing, all the groups will increase during the storage times, and 500 and 600 megapar has a lower value in day 15 compared to the others group. Table 5 shows the change of coliform in hard clam meats after HPP treatment during storage. The 0.1 megapar has detected of coliform from day 0 to day 15, while 200 megapar were only at day 0, and the other groups was not detected. This result shows that using 300 megapar and above can inactivate the growth of coliform during storage. Table 6 shows the change of viral parahemolyticus in hard clam meats after HPP treatment during storage. There were only detectors of VP in the control groups 0 from day 0 to day 6, and the 200 megapar were only at day 0, and the other group was not detected. The results of control groups at 200 may be due to the storage temperature, which caused the inactivations of viral parahemolyticus. Figure 9 shows the change of pH value of clam meats after HPP treatments during storage. As the result shows, the control groups and 200 megapar have decreased during the storage, and the others group will also decrease, but they will gradually increase starting from day 9 to day 15. Figure 10 shows the change of total volatile basic nitrogen TBN of hard clam meats after HPP treatment during storage. The dashed line represents 25 mg per 100 grams of TBN as the regulatory standard for raw frozen seafood. The control groups will have exceeded the standard at day 9, and 200 to 400 megapar has exceeded the standard at day 12. While during 15 days of storage, the 500 megapar and 600 megapar did not exceed the standard. And in day 15, the control groups had reached the highest value, which is 86.1 mg per 100 grams. Conclusion. APC, PBC, HBC, coliform, and VP of hard clam samples decreased with the increase of pressure. HPP treatments at more than or equals to 400 megapar has a bad shocking effect, and the expanding rate and moisture content will significantly increase as the pressure increased. The value of L and W significantly increased with the increase of pressure in 5 minutes, whereas value A and B will decrease. The APC and PBC levels in more than 400 megapar samples were retarded as compared to untreated samples and the less than 300 megapar samples during storage. The growth of coliform and VP and TBN contents in HPP treated samples were inhibited or retarded. In conclusion, the HPP treatment hard clam stored at refrigerations could prevent the quality deteriorations and extend the shelf life. My presentation will stop here. Thank you very much.